The first speaker to give the lecture is the laureate in advanced technology, Dr. Robert Samuel Langer. Dr. Langer, would you please come to the stage? Before we have his lecture, we will introduce the profile of Dr. Langer on the stage screen. Dr. Robert Samuel Langer was born in 1948 in the state of New York in the United States. He has two children, in other words, himself and his younger sister. As a child, Dr. Langer used to experiment with the erector set and chemistry set, which his parents gave him, and he gradually became interested in science. This is himself and his family. His father was very outgoing, his mother very kind. They raised their children with lots of love. In his high school days, he loved to play sports, and in the track and field, he won the gold award for the best track person. After studying at Cornell University, he went on to the Graduate School of Massachusetts Institute of Technology to major in chemical engineering. He wondered whether it was possible to make use of his knowledge on chemistry and engineering and apply it to the medical field. He wanted to treat diseases and injuries by using approaches of chemistry and engineering. In 1974, he became a researcher at the Harvard Medical School, and he started his research in order to achieve what he wanted to achieve. The third person from the left is Dr. Langer. At that time, the discipline spanning the fields of engineering and medicine had not yet been established, and therefore his research outcomes were not really recognized. But he didn't give up and continued to engage in research. He advocated a new concept of tissue engineering and contributed to the development of regenerative medicine. He used the biodegradable scaffold to culture cells, and by transplanting them into the body, it was made possible to regenerate the bone or an organ. This image is one of the applications of the drug delivery system of macromolecules like protein developed by Dr. Langer. The drug delivery system is the technology to deliver the necessary dose of the drug to the necessary site just when needed. With this, you are able to avoid massive dosing and alleviate side effects and reduce the time to treatment. Currently, Dr. Langer continues to engage in research together with the students and researchers who gather at his place, and he has launched a lot of startup companies and serves as biotech companies' advisors and still engages actively in the commercialization of the research outcome. The message to the young people is to dream big dreams that can change the world. Don't give up on those dreams and keep on going. That's his advice to the young people. This is a picture when the three children were still very young. This time, he is visiting Kyoto with his wife, and he says he looks very much forward to enjoying the beautiful city of Kyoto.
本日はランガー博士にご自身の開発者人生を振り返り、どのように苦難を乗り越え大きな発明に結びつけたのか、また開発者にとって最も必要なものは何かについて語っていただきます。演題は、ある The struggles and dreams of a young chemical engineer. それではランガー博士お願いいたします。
Now, Dr. Langer, please.
Thank you. What a wonderful honor it is to receive the Kyoto Prize in Advanced Technology. I feel particularly honored and humbled when I look at the accomplishments of the others who have received this prize this year and in previous years. I'd like to tell you how I got here today. It's not been a straightforward path. I grew up in Albany, New York. My father ran a small liquor store. He was very smart. I remember he would do crossword puzzles very quickly, and he had a very broad knowledge. He worked very, very hard, but when he came home, he would play math games with me. My mother, Mary, was a homemaker. She took care of my sister Kathy and I. My mother worried a lot. In fact, she still does. But she was and is one of the nicest people you could ever meet. I hope some of that rubbed off on me. Among the gifts my parents got me when I was between the ages of 10 and 13 was an erector set, a microscope set, and a chemistry set. I love these sets. I would make robots and rocket launchers with the erector set. I would watch shrimp hatch with the microscope set. And I set up a little laboratory with the chemistry set in the basement of our small house where I would mix chemicals together and watch them turn different colors. And I'd make things like rubber and polymers and other materials. I'd get other gifts as well, like baseball gloves, a basketball, and a football. I love these too, and I played a lot of sports with my friends in the neighborhood. I think I had a pretty regular and pretty normal, happy childhood. From the ages of 12 to 17, I went to the Milne High School in Albany, New York. I was very good at math, pretty good at science, and very bad at English and French. Both my father and my guidance counselor said I should become an engineer because I was good in math and science, and that's what I did. I applied to engineering schools, and I was fortunate to be accepted at Cornell University. In my freshman year, chemistry was my favorite subject by far, and it was the only course I did really well in, so I decided to major in chemical engineering. I worked very hard at Cornell, and I also got my first taste of teaching by being a teaching assistant in a course on heat and mass transfer. I loved being a teaching assistant. When I graduated from Cornell, I got several job offers to run chemical plants. But I didn't think I'd be very good at it, and it wasn't exciting to me. So I decided to apply to graduate school. I applied to 6 graduate schools, and I chose to go to MIT. But while I was at MIT, I did 2 things. The first is I did my doctoral thesis on enzymatic regeneration of adenosine triphosphate, also known as ATP. The goal was to explore enzymes for synthetic purposes as an energy source, and ATP was required. On the positive side of doing this, my advisor, Clark Colton, was very thorough, and one of the postdoctoral fellows in the lab, Colin Gardner, taught me how to do very careful, reproducible research. But on the negative side, after the first year or two, I began to think that the research I was doing wasn't really important. I couldn't see how it could have a great impact or how it might change the world. And this discouraged me about research. The second thing I did was get involved in a lot of educational outreach, particularly teaching underprivileged children. During my first year at MIT, I did a lot of tutoring in poor communities. And during my second year at MIT, some people wanted to start a school for poor high school children who had dropped out of the public high schools. They asked me to help, and we started what was called the group school. I helped create and chair the math and science departments. My big goal was to make math and science more interesting. I got MIT to fund a program to create a novel chemistry lab where we would teach chemical principles by practical examples. For example, making rubber illustrated a chemical reaction. Making ice cream illustrated freezing point depression. I also created a number of math games to help try to make math interesting. The school was very liberal politically, and the students were not required to take math or science. I remember when I first came to that school, only 5 of 40 students signed up for math. But 1 year later, 45 out of 50 students did, and that experience reinforced my love for teaching. I finished graduate school with a doctorate in chemical engineering, and I didn't know what I wanted to do career-wise. I graduated in 1974, and at that time, there was a big gas shortage. The price of gas went way, way up, and I lived in Boston, and you had to wait in line at the gas station for 2 hours to get your gas tank filled up. But the consequence of that is that if you were a young chemical engineer, you got a lot of job offers. In fact, nearly all of my classmates in the 1970s joined oil companies. They had many openings, and that's where the really high-paying jobs in chemical engineering were at that time. I received 20 job offers from oil companies, 4 from Exxon alone. I also got offers from Shell and Chevron and others. One job interview made quite an impression on me. I remember going to this interview at Exxon in Louisiana, Baton Rouge, and one of the engineers there said to me that if I could increase the yield of a particular chemical by about 0.1%, he said, "Wouldn't that be wonderful?" He said, "That would be worth billions of dollars." But I remember flying home to Boston that night thinking to myself that I really did not want to do that. What did I want to do? Well, from my college experiences, I had this dream of using my background in chemistry and chemical engineering to improve people's lives. And as I mentioned, I had spent a lot of my time as a graduate student starting a school for poor high school kids and developing new curricula in chemistry and math. And one day I saw an advertisement to be an assistant professor to develop chemistry curriculum at City College in New York. So I wrote them a letter applying for the job, but they did not write me back. But I liked that idea, so I found all the ads I could for an assistant professor position to develop chemistry curricula. I found about 40 such ads, and I wrote to all of them, and none of them wrote me back. So another way I thought I could help people was through health-related research. So I applied to a lot of hospitals and medical schools. They didn't write me back either. Then one day, one of the people, Barry Bunow, in the lab where I worked, said to me that I should write to a surgeon named Dr. Judah Folkman at Harvard. He said sometimes he hires unusual people. And Dr. Folkman was kind enough to offer me a job. So I took what at that time seemed like to all chemical engineers a huge risk and began doing postdoctoral work in a hospital. It might seem more common today, but at that time few, if any, chemical engineers had done postgraduate work in a surgery lab before. Dr. Folkman told me about a problem he had been working on, trying to figure out how blood vessels grow to cancer tumors in the body. He wanted to see if there was some way to isolate a substance that could stop the blood vessels from growing, something he called an angiogenesis inhibitor, because he felt that if you could do that, stop the blood vessels from growing, perhaps you could prevent nutrients from getting to the tumor, and that might stop the tumor from growing. When I started working with him, angiogenesis inhibitors were only theoretical, and many people did not agree with this concept. Moreover, the area of blood vessel growth in the body was very difficult to study. We realized that in order to solve this problem, we would not only need to isolate an angiogenesis inhibitor, which is often in the form of a large molecule, but has as often been the case in the development of new medicines, discoveries were hampered because no bioassays existed. So we would need to create a bioassay ourselves. To do that, we chose the cornea of a rabbit, because there are normally no blood vessels in that cornea. We put a tumor in the cornea in a way that would mimic what happens in the human body. And over 2 to 3 months, blood vessels grew from the edge of the cornea to the tumor. At that point, we wanted to stop the blood vessels from growing. But to do that, we needed a controlled release polymer that we could put in the eye that could deliver the different molecules I was isolating, and it had to be able to deliver them over a 2 to 3 month period. Since there were no such systems at the time, I tried to develop one. And my research in the drug delivery field started with this project. So the work I was doing was somewhat basic to help in angiogenesis research. However, from the standpoint of potential practical impact, something happened that I could not have anticipated. That was the advent of biotechnology and genetic engineering, where for the first time it became possible to create large molecules, such as peptides and proteins, in a commercial way. But these molecules face serious delivery challenges. Swallowing them did not work because they were too large, and they'd be destroyed by enzymes and acid in the stomach or the intestines. They were also too large to put in a transdermal patch. And if you injected them, they were quickly destroyed by enzymes. But delivering any of these molecules on a chronic basis would require a way to deliver them in an unaltered form and yet protect them from harm. When we started this work, the conventional wisdom in this field was that this could not be done. Scientists simply felt it was not possible to slowly release large molecules from biocompatible polymers any more than any person could walk through a wall like that. Against this background, I began working in the laboratory to see if I could come up with a way to make tiny systems that could deliver these molecules for a long time. Over 2 years of experimentation, I found hundreds of different unsuccessful methods. But finally, I discovered one way to make it work. My students and I took water-repellent polymers like ethylene vinyl acetate copolymer or lactic glycolic acid copolymer and dissolved them in organic solvents, usually at very low temperatures. We added the molecules to them, slowly dried off the solvent, and this enabled us to create small microspheres, or, as I will tell you later, even nanospheres. We published this in the journal Nature, that you could use this approach to release molecules of almost any size. The molecules could be released for more than 100 days in test tubes. Although the release rates were not constant in these initial studies, we later developed ways to ensure constant rates. Here is an example. At first, our concepts were not well accepted. Scientists and engineers ridiculed this work. For example, when I was a postdoc, I had not realized that being a member of the scientific community meant giving speeches at conferences. In 1976, I was asked for the first time to give a lecture at the Midland Macromolecular Symposium in Michigan. I was 27 years old at the time, addressing distinguished elder polymer chemists and engineers. I'd never given a big talk before. In fact, the only talk I'd given before that was in 8th grade, when I had to give a 90-second speech. So the night before my 8th grade talk, I rehearsed that talk for 4 hours in my parents' bedroom in front of a mirror. The next day, I started to give the talk, but after a minute of speaking, I could not remember the next word, and I froze. I'll try not to do that today. And eventually, the teacher told me to sit down and gave me a very poor grade. I think it was an F. So now, when this Michigan talk came about many years later, I was very nervous. So I stopped working about 2 weeks in advance of the talk, and I kept practicing the talk over and over in a tape recorder until finally the day came when I was going to give it. I got up, I gave the talk, and at the end of it, I was pretty pleased because I didn't forget too much what I was going to say, and I didn't stammer too much. And I thought when I was done with this talk that all these much older, distinguished chemists and engineers in the audience, being nice people, would want to encourage me, this young guy. But when I was done, a number of the people gathered around me and stated, "We don't believe anything you just said. We know you can't get those molecules you're talking about to slowly diffuse through polymers." It wasn't until several years later, when other people began repeating what we did, that the question shifted to how could this possibly happen. In fact, I spent a good deal of my early career at MIT understanding how these polymer drug delivery systems functioned and tried to make them useful for different applications. Also, shortly after this talk, I tried to receive funding to support my research, and I wrote a number of grants. My first 9 were turned down. I remember I wrote one grant to the United States National Institutes of Health, and I got the review back, and they were very, very negative. They not only turned me down, but they said, "Well, how could Dr. Langer do this? He's a chemical engineer. He doesn't know anything about biology, and he knows even less about cancer." When I was done with my postdoctoral work, I applied for faculty positions in a number of chemical engineering departments. But I had trouble getting jobs because people felt at that time what I was doing wasn't engineering. They thought it was more like biology. So I ended up joining what was then the Nutrition and Food Science department at MIT. But in that department, what happened was the year after I got that position, the chairman of that department who hired me left, and a number of the senior faculty in the department decided to give me advice. And their advice was that I should start looking for another job. So there I was, getting my grants turned down, people not believing in my research, and it appeared that I would not even get promoted to associate professor. But fortunately, within the next few years, scientists in the pharmaceutical industry and different universities started using some of the principles and techniques I developed, and things began to turn around. Also, going back to our research in the 1970s, people couldn't understand how we got long-lasting release. To answer this, Rajan Bawa, one of my students, began to use a cryomicrotome, a low-temperature cutting instrument, to cut thin sections through one of the polymers we used to visualize it. So we did a permeability study on a very thin slice of polymer, and it looks like this. We found that if molecules were 300 molecular weight or greater, they wouldn't be able to diffuse from one side of this to the other. So how could release happen? Now, to understand this better, we put a reddish molecule in the polymer, and this is before any release. Now we began the release process. By the way, notice the two separate phases: polymer, drug. After we did release for a year, we cut another thin slice and found that what was left behind where the reddish molecule was were these holes. Since these holes did not exist before the addition of molecules, they clearly had been made by the way we made the polymer systems. And we did a lot of imaging and even scanning microscopy studies and found that these holes were interconnected. They also had very tight constrictions between them, and they were incredibly winding and tortuous, which accounts for the very long time it took the molecules to get through. I sometimes like to explain this long release process by comparing it to driving a car through my hometown of Boston, which is often very tortuous. We then discovered that by adjusting these porous structures, we could actually make tiny systems that could last anywhere from a day to more than 3 years, or any time in between. One of my goals in doing laboratory work has been to move beyond just conducting the work and publishing the results, but also applying that work to helping people. The first thing I did to work with Dr. Folkman was to use this polymer system in the bioassay I mentioned earlier to see if we could find a substance that could stop blood vessels from growing in the body. I had isolated many different substances, and I tested all of them in over 1,000 different studies. Every substance but one failed to stop the blood vessels from growing, but one of them did. We did over 20 different experiments with the substance, and it was very exciting. For the first time, we saw that blood vessels growing towards a tumor were halted. They actually formed a zone of inhibition around the polymer that was releasing the inhibitor. This had never happened before in thousands of experiments. We published this paper, which showed that for the first time that inhibitors of blood vessels did exist, and it also provided bioassays which could and were used to isolate all future inhibitors. We published this in the journal Science in 1976. Today, many blood vessel inhibitors have been isolated. They have been used to treat over 20 million patients who have different types of cancer or eye diseases, such as macular degeneration. Regulatory authorities, like the United States Food and Drug Administration, now say that there are 4 ways to treat cancer: radiation, chemotherapy, surgery, and stopping blood vessel growth. That's called anti-angiogenesis. And of course, many times, several of these therapies are used together. After our experiments worked successfully in that situation, I wanted to move to the next logical step, which was getting the drug release systems to work through the body in animals. For this experiment, I worked with one of my graduate students, Larry Brown. The model system we chose was diabetes. Our goal was to deliver insulin, which is a large molecule, to diabetic rats to lower their blood sugar. So after we made the rats diabetic, we made small plastic pellets, no bigger than a tiny pill, that contained insulin, and put them into rats. We found that the pellets could lower the rat's blood sugar from 400 milligrams, which is diabetic, to 100, which is normal, for over 100 days. And this experiment showed us that we could maintain biological activities of molecules like insulin for long periods of time. Of course, my long-term goal was to see if we could get this drug release process to work in humans, and I'll talk about that more later. The original controlled release materials we developed were small particles, but in many cases they are microspheres that you might inject under the skin to release molecules for long periods of time, like weeks or months. Another challenge, though, is controlling the location of the drug. To solve this, we even created nanoparticles that you could inject into the bloodstream. Nanoparticles are critical for delivering significant amounts of any drug into cells, particularly newer potential drugs like siRNA that can shut off genes. But polymer nanoparticles injected into the body are normally destroyed almost immediately by certain cells called macrophages. This made their use essentially non-existent. In a 1994 science paper, we addressed this and created nanoparticles that could circulate for hours or days in animals, be stable on the shelf, and not aggregate. Let me show you a video.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should point out, a lot of the medical doctors I work with tell me the cell doesn't get blown up quite that way. These principles, however, are now being widely used by many scientists and companies to practice the new area of nanomedicine. Another area I started thinking about involved creating new polymer materials. Working in a hospital, it became clear to me that almost all polymers used in medicine were derived from household objects, and their medical use was driven by clinicians who wanted to use them in medical areas based on their physical properties. For example, the polymers used in a lady's girdle are now used in artificial hearts because they had a good flex life. The polymers used in mattress stuffings are now used in ladies' breast implants. Yet such an approach often leads to problems. Artificial hearts, for example, can cause clots to form when blood hits their surface, the lady's girdle material, and these can cause clots, which can cause strokes and death. Against this background, I began thinking that we needed to find ways of solving medical problems other than to search for materials in everyday settings. As a chemical engineer, I believe that researchers could take an engineering design approach by asking the question, what do you really want in a biomaterial from engineering, chemistry, and biology standpoints, and then synthesizing these materials from first principles. And as a proof of principle, we decided to synthesize a new family of degradable polymers, polyanhydrides, for medical use. The first step was to select the monomers, the building blocks of the polymer. I asked Michael Marletta, who was then at MIT, what monomers, from a list of substances we thought might make good polymers, he thought would be safe in the human body. We then synthesized these polymers and discovered that by changing their compositions, we could make them last anywhere from a day to many years, or any time in between. Then, with Henry Brem, who is now chief of neurosurgery at Johns Hopkins, we thought we could use this polymer to locally deliver the drug to treat brain cancer. But I had to raise money for this project, so I wrote grants to the National Institutes of Health and other funding agencies. These grants were then reviewed by what are called study sections composed of other professors at other universities. And their reviews were very, very negative. In our first grant, in 1981, the reviewer said we would never be able to synthesize these polymers. However, Howie Rosen, one of my graduate students at the time, synthesized these polymers for his thesis. He eventually became president of the Alza Corporation, a very successful company, and was later elected to the National Academy of Engineering. After Rosen accomplished that synthesis, we sent the grant back for review and received the reply that it still should not be funded because the polymers will react with whatever drug you put in. But Cam Leung, now the James Duke Professor at Duke University and a member of the National Academy of Engineering, and Bob Linhart, who also is a chaired professor at RPI, they were postdocs at the time, showed there was no reaction. We returned the grant for another review, and it came back with the comment that the polymers were fragile, they'd break. This time, two other postdocs in our laboratory, Avi Dome, who later became chair of medicinal chemistry at Hebrew University, and Edith Mathiowicz, who's now a full professor at Brown University and has been elected to the National Academy of Inventors, addressed this problem. The revised grant was sent again for evaluation, and this time the reviewer said that it should still not be funded because the polymers would not be safe on animals or people. But I had another graduate student, Cato Lorenzen. He would eventually become Dean of Medicine at the University of Connecticut, a member of National Academy of Engineering and the Institute of Medicine of the National Academy of Sciences, and he did show they were safe. These kinds of negative reviews occurred for a long time, but in 1996, the regulatory authorities, like FDA, finally approved the treatment. It was the first time in over 20 years they approved a new treatment for brain cancer. The first time they ever approved this idea of polymer-based local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well the graduate students and postdocs have done. They have become heads of major corporations or very successful professors at top universities. I should add that the funding that we obtained to support our research was based on patent coverage on polyanhydrides that we received from a company that licensed our patents. 18 years after their approval, these polymer systems are still used for treating brain cancer in over 30 countries, including Japan. They're marketed by Asahi in Japan, actually, and they provide an entirely new paradigm in the drug delivery field, helping pave the way for drug-eluting stents and other delivery systems. I would now like to describe another area, the delivery of mammalian cells through materials, which our group has been working on for some time. My motivation for working on this project goes back more than 30 years to work we did with Boston surgeon Jay Vacanti, who's now chief of pediatric surgery at Mass General Hospital. Among his patients were small children dying of liver failure whose lives depended on someone else dying and donating a liver for transplant. But of course, not enough transplants are available to treat these patients. And this problem is not limited to liver disease, but it extends to pretty much every organ or tissue in the body. Over 30 years ago, Jay and I came up with an idea for tissue engineering. The concept involves taking isolated dissociated cells from a patient or close relative. These are some of the different cell types. And they could be bone or cartilage or liver cells. They could also be stem cells. If the cells are injected at random, very little happens. But it turns out that these cells are smart. And if you put the cells close enough together, they can organize themselves and create structures. A group, for example, at University of California at Berkeley, has shown that if you take mammary epithelial cells and put them close enough together, even in a test tube, they'll form what are called acini and make milk. We wondered if we could make tissues by taking cells and putting them together on a polymer template in 3 dimensions with the right media. We would grow the cells outside the body on the right materials and then ultimately return them to the body to make whatever tissue we wanted. And we would use either existing FDA-approved polymers or synthesize brand new ones. Let me speculate where this field might be in, say, 30 years. A good example could be plastic surgery and asking for a new nose. I believe in 30 or 40 years, patients might be able to go to a computer screen to select whatever nose they might like or create their own designer noses. Prasad Shastri, one of my postdocs, now professor at the University of Freiburg, worked out a way to do just that using a novel fabrication process that includes three-dimensional printing that could be someday adapted for computer-aided design techniques. He was able to fabricate a regular nose from a polymer, and it's 97% porous. Pieces of this polymer could be added or reshaped to create other kinds of shapes or noses that the patient might desire. The donor cell for the nose could be taken, for example, from the patient's ear through a minimally invasive procedure called an arthroscopy. They could be placed on the scaffold until the nose is fully formed. And we're now using this procedure, as I'll go over, but not on noses, as I will describe shortly. And let me, along those lines, give you 3 examples of tissue engineering using the principles I've discussed. The first one is in the form of replacement cartilage. Every year, about 1 million people need new cartilage. So we did some experiments on nude mice using their own cells as a scaffold to develop new cartilage. We even reconstructed the skull of a mouse and the cheek of another. And if you opened them up, it looked like pure cartilage. However, this replacement cartilage we made is not as mechanically strong as you want it to be. So we're still working on it. So we're really not able to help, say, runners yet, but we can help people that have certain cosmetic defects. Jay, my colleague, sees patients who sometimes don't have ears. And Jay, Linda Griffith, and I received funding from the United States Army to help create body parts for ears for soldiers who come back from Iraq and Afghanistan without such body parts. We create a human ear by making a scaffold in the form of an ear with polymer fibers, cartilage cells, and while this has not yet been tested on humans, it has been tested on rabbits. And it's even been used on patients. Here's an example of a 12-year-old boy. Notice, however, he has no chest covering his heart. And like other 12-year-old boys, he likes to play baseball. But if he were ever hit in the heart with the baseball, he could die. And Jay operated on him and gave him a new chest, and we created a polymer scaffold for him. Now it's many years later, and this boy is a grown man and is doing very well. A second example is the development of new skin for burn victims. Let's consider an actual patient where this was done, a 2-year-old boy who was very badly burned. The clinicians took a polymer scaffold with new neonatal skin fibroblasts. They put it, here it is, they put it in the child at the time of the injury, but 3 weeks later, he looks like this. 6 months later, the burned area is gone. And this treatment has been now approved for patients with skin ulcers and burns. And one other example I wanted to give you is spinal cord repair. Erin Levick, who is now a professor, she was a graduate student, she's now a professor at Case Western Reserve. For her thesis, she made a polymer scaffold that mimicked the human spinal cord. And she created an outer portion to help guide axons, an inner part where we put neuronal stem cells. And we then worked with a neurosurgeon, Ted Tang, and a stem cell specialist, Evan Snyder, to test this in animals. And we had paraplegic rats that we made, and we actually, they had trouble moving their hind limbs. We divided the mice into 4 groups, a treatment group that had the polymer cell scaffold inserted, and 3 control groups. One just had a sham operation, one had stem cells only, and one had polymer only. We then observed the rats for a year, doing numerous studies. And at 100 days following the operation, we found that in the control groups, all the animals dragged their legs. Let me show you a video. So this is a paraplegic animal, and we had about 40 of these in the control group. Notice the paws are splayed in an awkward fashion, and he's not able to support his weight. This is typical. But now, if we put the scaffold with the neuronal stem cells in, it's dramatically different than the control groups. These rats can even bear their own weight, even though they're heavy, and they can walk. The paws are splayed in a much more normal fashion. These studies were repeated by neurosurgeons John Slotkin and Eric Woodard and scientists at In Vivo Therapeutics in non-human primates, African green monkeys. And recently, the FDA gave the go-ahead to start human clinical trials, and the first human patient was treated just 3 weeks ago. I also very much wanted to have the inventions and materials that we developed get to the point where they could help patients. And this is difficult because it takes a great deal of money and people to develop medical products. So what I did was to begin writing patents on our work. We have over 1,000. Today, we've licensed and sub-licensed these patents to over 300 different companies, and I even began helping to start some of those with my students. I should add that when I wrote these patents and helped start these companies, many scientists and professors looked down on it. They thought this was not a very good thing for a professor to do. But today, these companies have made all kinds of products that treat patients with cancer, heart disease, and many other sicknesses. These companies have also created many, many thousands of jobs throughout the world. Let me give you a few examples. In the first case, let me expand on what I mentioned earlier, when we developed polymer systems that could continuously release large molecules. One of the challenges was getting a patent. In fact, we filed a patent in 1976, and it was turned down. It was turned down actually 5 times between 1976 and 1981. And my lawyer said we should just give up. But I don't give up so easily. And I started to think of new ways we could get the patent allowed. The patent examiner said that what we had done was obvious. But I knew that wasn't true because, as I mentioned before, scientists kept saying it was impossible. So I went to the literature, and I discovered that a paper published by 5 famous chemists and chemical engineers 3 years after we wrote our paper in Nature, and they referred to our work by saying the following. Generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and Langer, myself, have reported some surprising results that clearly demonstrated the opposite. So I showed this to our lawyer, who said, "Oh, this is very interesting." And he then showed it to the patent examiner, who said, "I will allow this patent if Dr. Langer can get what are called written affidavits, written statements from each of these chemists and engineers saying they really wrote that in an article." And all 5 scientists were kind enough to write me back that they did write that, and we got this very broad patent. We then licensed it to 2 very large companies, one in animal health and one in human health. And both companies gave us grant money, and they promised to do experiments to develop our invention. And I was so excited about that. These companies, however, were very large. And so what happened is they do 1 or 2 experiments, and then when they didn't work so well, they gave up. So a few years later, Alex Klebanoff, one of my colleagues, said to me, "Bob, why don't we start a company ourselves?" So I was able to get these patents back, and we started a little company called Enzotech, which later merged to become Alkermes. And Alkermes has made all kinds of these long-acting microspheres that have helped millions of patients that have suffered from diabetes, schizophrenia, alcoholism, narcotic addiction, and pituitary dwarfism. In a second example, we started Momenta Pharmaceuticals with two of my graduate students and postdocs, Ram Sesasekaran and Ganesh Venkatranam. Here, the technology that we created involved enzymes called heparinases for polysaccharide sequencing. And this was described in papers in Science that we wrote, and again, protected by patents. This technology is being used to create new anticoagulants and other drugs for use in heart disease, cancer, and other diseases. Momenta also used this sequencing approach to create the first generic low molecular weight heparin, which is an anticoagulant. And because the sequencing confirmed the precise chemical identity of the heparin compared to the original heparin, we were able to get what's called an abbreviated new drug application approved by FDA, saving many millions of dollars and many years of research that are normally used for these molecules. Today, this product has again treated about 10 million patients, and it was the largest syringe launch in history. A second product is pending approval at the FDA, and others are in clinical trials. But furthermore, in 2008, there was a heparin crisis in which contaminated heparin from China killed hundreds of people in 22 countries. Ram and I, as well as Bob Linhart, another of my former postdocs, work with Momenta, the FDA, the Center for Disease Control and Prevention in the United States to sequence the contaminated heparin, identify the contaminant, set new standards. And after we did that, there were no more deaths. The above companies are just examples of how we've been able to move discoveries from the lab to patients. We've been involved in launching some 30 companies, and they make all kinds of things. New aerosols to treat diseases like diabetes or Parkinson's. Doing new types of genetic therapy for treating diseases like cancer or heart diseases. Developing new types of drug delivery microchips that may someday enable personalized medical therapy for treating osteoporosis or other diseases. Creating new therapies for spinal cord injuries, I mentioned. Create new nanotechnology approaches, both therapeutic and diagnostic. And even new hair and skincare products. Polaris and Ventures and the Chemical Heritage Society estimates that over 2 billion patients will be helped by these products created by these companies. I wanted to just tell you a little bit about my personal life. I've been married for over 26 years to a wonderful, beautiful, and kind woman, my wife, Laura. She's the attractive one on this slide. I met Laura because she was the roommate of one of my postdocs, and I'd seen her running on the track where I also ran for exercise. As I said, I thought she was very beautiful, stimulating, smart, and nice. Laura has a bachelor's degree from Harvard and a PhD in neuroscience from MIT. Being married to someone with a scientific background has the added benefit that she knows the pressures I feel and the rewards I get from science, and being able to share that with her has been wonderful. Laura is my best friend, as well as my wife, and I've been very lucky because Laura is also a very straightforward person. I always know exactly where I stand. I should add that a number of years ago, my postdocs and students had a symposium for me, and Edith Mathiowicz, one of my former postdocs, made a graph of my productivity. And she asked everybody in the symposium a question. She said, why do you see the sharp inflection point here? She even made an equation out of it at 1986. And she said, the answer is that's when I met Laura. We also have 3 wonderful children, Michael, Susan, and Sam. This is the picture you saw earlier, but let me just show you them today. That's Michael, Susan, and Sam. I feel very, very lucky because being a husband, a father, and a scientist has enabled me to have a very complete and really a wonderful life. Throughout all of this research, with its challenges and setbacks, I remain incredibly excited about the potential for science and engineering to make the world a better place and transform human health care. I also believe we are only at the tip of the iceberg in creating materials for all kinds of applications that can profoundly relieve suffering and prolong life. I've also learned a great deal from the challenges and setbacks I faced. And if there's any advice I might give to young people, it would be to dream big dreams, don't give up on those dreams, and recognize that conventional wisdom is not always correct, meaning that what you read is not always correct. I feel that I've been incredibly fortunate that I've had such a wonderful staff at MIT with such super students, postdocs, and collaborators. I've seen over 700 of my students get wonderful jobs. Over 260 are now professors around the world, including a number who are professors in Japan. I view my students and postdocs actually as an extended family, and I'm so very proud of them. I would not be here today without having had so much support and so much help from so many people. Once again, I'm tremendously honored to be chosen as a Kyoto Prize recipient and to have had the wonderful opportunity to share my thoughts, struggles, and dreams with you this afternoon. Thank you very, very much.
Thank you very much. We would like to present a bouquet of flowers to Dr. Langer.
ランガー博士にもう一度大きな拍手をお送り。
Please give a big hand once again to Dr. Langer.